the entire 2 hours. Let's see how it goes.
Okay, it should be.
Do I just go to the other screen? Or which screen do I actually go to?
Yeah. I'll just check it. Wait.
Oh, it's okay. I think you usually don't have issues with it, so. And, right, thank you.
All right, perfect. No problem. Okay, good luck. See you.
Would I be able to record the lecture?
Sure, that'd be fine. I'm going to as well.
You're what, sorry?
I'm going to as well. So
Oh,
we'll
okay.
see how it goes.
Okay. Would I be able to slide this open?
Sure. If any of you need a hard copy of today's lecture, I have the set printed out for both today as well as next week in case you need a copy. There are about 6 or 7 in front.
Okay, if you don't mind, I'm going to try to get going. I am going to try recording the lecture that we're going to have both today as well as next week. If you want the link, we're going to be streaming actually directly onto YouTube. And if you want the link for that, I'll be happy to pass it along to one of your classmates. And you can try to follow along with that as well in case you miss a point or if you want to go back and review in a little bit more detail. So I'm going to take you through the digestive system, which in many ways is a really simple system. I'm more interested in you learning the material rather than you trying to memorize the material. So wherever possible, if you have a question, feel free to ask. I'd be happy to try to answer it. I want actually this to be more of a real world physiology because that's why you're here studying physiology, right? Because you want to make sense out of an actual physiological process that you're used to, that you do every single day, or you should be doing every single day. And hopefully by the end of this lecture, you'll have an idea about some of the basics, the anatomy of the plumbing that goes through your body. In many ways, it looks a lot like that diagram I showed you at the very beginning. And actually, I'm going to be talking to you about the GI tract overall today, and then we'll come back and revisit a couple of things in the next lecture. I want you to have a good idea as to why you can't control the GI tract, and we'll go through that in a few moments. And at the beginning of the next lecture, I'm going to tell you why you actually have to eat, and the only reason that you actually eat is because you breathe. If you didn't breathe, you wouldn't actually have to eat. Not because you're dead, but because you actually don't need something as part of the respiratory system, as part of the oxygen sink. So I'm going to go through a couple of different things with you here today. I'm going to go through a couple of aspects of the GI tract, some of the things that I think are really important for you to understand as a basic functional unit, and we'll define a couple of things related to some of the basic structure as well as the function of the GI tract and digestive system. And throughout this entire lecture series, one of the things that I want you to be aware of is that the GI tract system really is a very hostile environment. It resides inside of you, so you don't really think that it's a hostile environment, but it actually has to be in order for you not to get sick. Every time you're eating something, whether you think it's sterile or not, you're actually ingesting bacteria, and that bacteria has direct access to your body through your GI tract system. If there wasn't a way to destroy that in a very effective and efficient manner, you'd be in trouble really, really quickly. And so we're going to be looking at a number of different factors, including how there are different pH changes in different parts of the GI tract, and it all actually fits together. Different pH changes in different parts of your body make sense because of different enzymes that are at work. And in addition to that, we'll talk about some of the different changes in the different muscle layers that line the entire tract. From the beginning right to the end, it's a muscular tube. And this muscular tube undergoes a number of different changes along the way. Both of these things, the muscular tube that I just mentioned, as well as the enzyme system, really tell you that there are mechanical forces, the muscle part, and then the chemical changes, which involve the pH changes that I mentioned earlier. So we have chemical digestion going on as well as mechanical digestion going on. Most of all, though, I want this to be relevant for you. You eat, and you have to think a little bit about the whole process. Like, why do you bother eating? What sorts of things do you actually eat? And how does that actually get digested in meaningful ways? And I want it to be relevant for you as an urban biologist. So we're going to go through in the first few parts of this lecture into the different layers of the GI tract. You'll learn, hopefully by the end of today, the 4 basic layers that make up the GI tract wall. It is the same throughout the entire GI tract system and the digestive system that we're going to be looking at, and then some of the different chemical and mechanical systems that go on throughout the length of this GI tract system. I also am going to talk to you about some of the major accessory organs. So if you're thinking about the GI tract system sort of being line 1 on the subway line, some of the other lines also connect and make connections with this line 1, and these are some of the accessory lines that we're going to be talking about and the accessory organs. They all have to work together, though, in order for digestion to work really, really well. Ultimately, by the end of next lecture, not today, but at the end of next lecture, you're going to understand that we break down food, the macromolecules that you take in, the basic food components that you take in. We can't digest that. If you were to take your hamburger and place it up against your small intestine, no matter how long you left it beside the small intestine, none of it would get absorbed. In fact, it has to undergo a series of stepwise reactions in order to make it smaller and into definable units that we call digestion in order for it to actually be absorbed into the small intestine. And we're going to be covering all of this in the upcoming lectures over the next 2 weeks. And again, as I mentioned, I want it to be relevant for you. So I want you to ask questions, whether it's through email or through dropping by my office, which is on the other side of campus. Feel free to let me know what sorts of things are really interesting for you. When we start talking next week about absorption of some of the digested, the chemically broken down, mechanically digested food products that we're going to start into today, this is how we gain weight. You know, none of us really go out and start thinking that, you know what, I want to eat this food just to gain weight. Unless you're a professional athlete, unless you're a professional food eater, you don't have that intent to begin with. But some of these things that we're going to be talking about have some social implications as well. So as I mentioned to you at the outset, and you don't have to worry about any of the diagrams or anything else so far, I'm hoping it's just an overview for you. You don't have to memorize anything. I'll tell you exactly what I want you to take away. I really want you to learn as much as possible, or to remember as much as possible if you've learned this before. When we talk about the digestive system, it's really this red line that we're looking at, where it's running from the esophagus all the way through your stomach, small intestine, and then out through the large intestine. That's the digestive system. That's the muscular tube that we're going to be talking about. Sometimes it's a little bigger, sometimes there are more muscle layers, but overall it's the same system all the way through, and that's why it's the same color all the way through. And you'll also see that we have accessory lines that feed into it, things that are important to help in this digestive process. And these accessory organs are ones that we're going to point out, make sure that you understand their major functions and how some important accessory organs we actually couldn't survive without. Okay, so we're going to talk about the major sections, starting from the mouth, the part of the pharynx, into the esophagus, the stomach, small intestine, large intestine, and then right down to the anal canal. And we'll be finishing off sort of at the level of the large intestine today. And then next week, we'll talk about absorption. Do you have any questions about anything so far, or anything that you'd like to talk about next week? No? Are any of you feeling particularly adventurous right now? It's not an adventurous class. It's a really shy class. Just going to sit there? Yeah, pretty much? Okay. I need someone to give me a hand with this. Anyone want to give me a hand with this? Yes? Okay, great. At least one adventurous person here. I have a little bit of a cold, so I'm going to keep it in the wrapping, and if you could help yourself, and feel free to do whatever you wish with it, but hopefully it involves doing something that we're learning about in class today. So we're also going to be talking about some of the major accessory organs of the digestive system, and these accessory organs include things like the salivary glands, the liver, the gallbladder, as well as the liver biliary system. They're put together, and then the pancreas. And in fact, I'm going to mention to you right now, the pancreas is one of the most important accessory organs that we actually have. If you're looking at all of these together, and based on the anatomy you've already had in class, can you tell me anything that's common with all of these different organ systems? Yes. They're secretory organs, meaning what? They all tell secrets to each other? And what do they secrete into? You're absolutely right. Sorry? Into the GI tract through what system? The vasculature. So that would be more endocrine. But these use ducts, and these are all duct system based. So they produce substances. We're always going to see there's a duct associated with them. There's a pancreatic duct. There's a hepatobiliary duct system. Salivary glands also secrete into ducts. These are all related to ducts. They're not related to the endocrine system, but thank you. I really appreciate your answer. And so we're going to be working through each of these different processes. Some of these, as I mentioned, are actually under your control. So your colleague who is deciding not to eat at this time, trying to get it open. Okay, so at some point, that eating process is completely 100% voluntary. You control, like the chewing, that's something that you have to consciously do. At some point you have to swallow, but beyond that, once you get past this mechanical processing and then you get into digestion, everything else is completely out of your control. And these are important aspects when we get into thinking about the digestive system and the autonomic control of the digestive system as we go along. So far, I've talked a lot about the overview of what we're going to be covering, why we're going to be covering the different aspects of the GI tract system. We're waiting for your colleague to actually start eating, start digesting, start enjoying, and we're going to catch up with them a little bit later on. So if you're thinking a little bit about this, I told you at the very outset, we are looking at line 1 today. We're looking at that red line. We're looking at that line that runs from the esophagus all the way down to the anal canal. It has some modifications along the way. But overall, it's a muscular tube. I told you, without going into detail, that it's going to involve mechanical digestion. There's going to be movement. There's going to be things kind of sloshing around in there. In order for you to be thinking about this, though, if you are thinking about this, if that tube is running through your body the way that I suggested that it does, there has to be something that actually makes sure that when your intestines, or when your large intestine or small intestine is actively contracting and moving around, there has to be a system that doesn't allow it to get entangled or twisted on itself. And the next sort of few slides, the ones that I do want you to pay attention to, the ones that I want you to focus a little bit more, this mesentery system is like a series of drapes. They're drapes that we don't really think about. Most of the time when you're thinking about the digestive system, you're just thinking about this tube system, as I mentioned to you earlier. But if it is really moving around that much, if it's collapsing on itself and as it's causing and pushing food to move from one end of the tube to the other, we need to make sure that it's not moving around too much. And this mesentery system that I'm going to show you in the next few slides is really important to keep the different parts of the system in its appropriate place. And whenever we have an organ filling a particular space or a cavity, and in this case we call it the peritoneal cavity or the abdominopelvic cavity, they're really the same thing, that when we have an organ and series of organs filling a cavity, we need to wrap them in mesentery. It's the same thing with your lungs, it's the same thing with your heart. We just call this mesentery different names. So this mesentery, which is surrounding the organs that are part of the peritoneal cavity or the peritoneum, is this membranous sheet. And it's found within this abdominal cavity, so right around here, so right under sort of where your liver is at, all the way down to the pubic symphysis. This abdominopelvic cavity is lined with a serous membrane. So it secretes fluid, in other words. Whenever you see the word serous, it means that it's fluid secreting. And you need that. You need that serous membrane that secretes serous fluid because your organs are moving around. There has to be some lubrication in place. And in fact, it secretes enough that it actually generates what's known as peritoneal fluid, and it provides that lubrication so that the different organs and the different accessory organs that are all packed together within this abdominal cavity, as you're moving things around and as you're digesting them, actually don't rub up against one another, they don't encounter friction, they don't twist on top of each other, they maintain their overall position. And this is what it kind of looks like. This is your standard textbook view of what the abdominal pelvic cavity or the peritoneal cavity looks like. But if you're actually taking a look at it from the side view, if you were to take a look at how, for example, the stomach is kept in place, there are a series of these mesenteries, those sheets, the double membrane sheets that secrete fluids in this peritoneal space that actually maintains the overall spatial location of the stomach where it's supposed to be. It's going to be contracting a lot. It's going to be mechanically digesting food a lot. So are the different parts of the colon. They're going to be moving around a lot as well, but they don't move too far because of this mesentery system that we have in place here. Okay, so these help to suspend different parts of the digestive tract from different parts of the peritoneal cavity. And the next slide is the one that I want you to be focusing on just a little bit. So you might want to circle it in your lecture notes, or you might want to fold your lecture notes over to come back and review this in terms of the anatomy if it's something you haven't encountered before. So a lot of these different structures that I've just shown you are actually hanging off of other organ systems. So we have like one sheet system here, and we have these other systems here. And some of these different sheets collapse into what are known as ligaments. And one of these ligaments that's really important in maintaining the overall structure relationships, like how one structure is in relation to another, is known as the falciform ligament. And it's important because this mesentery sheet, which has collapsed down into a ligament, so it's like having your drapes being tied together and then suspending from one part of the curtain rod down to another part of your home. It attaches from to the liver from the abdominal cavity wall. So in other words, it helps to maintain the liver in its normal position. Okay. There's another structure called the lesser omentum, and it's also part of that mesentery sheet system that joins the liver, stomach, as well as the duodenum, which is the first part of the small intestine. There is another greater omentum, which is just another name for a different part of the mesentery, and this is called the greater omentum because it covers most of the other parts of the small as well as the large intestines. Hopefully that's not anything too new for you. If it is, again, I want you to go back, maybe over the weekend, just go back and review and make sure that you are aware that all these different structures that are moving around during digestion are kept in place by these series of sheets or these ligaments that help to keep them there. They're not only there, though, to maintain the overall structure relationship, to keep one structure from moving around behind another structure or over top of another structure. They're also there because, for example, when you're gaining weight and when you see where fat gets deposited, fat doesn't just randomly get deposited, for example, just anywhere in your abdominal cavity. It starts off, the adipocytes, those fat cells actually start accumulating here on this sheet, on this greater omentum. So these different structures actually play really important roles. When you're gaining weight, when your adipocytes are accumulating and you're causing that beer belly formation, they're accumulating first at the level of the greater omentum, which covers those small and large intestines. And again, if you're getting an infection in your abdominopelvic region or the peritoneal cavity, those infections often start here at the level of the greater omentum, which is why, for example, you often get infections of the large intestine or the small intestine rather than infections of your stomach, etc. Okay, so do you have any questions about that before I go on? Is that somewhat of a review for you, hopefully? Have you ever heard of omentum before? Think of it like momentum, but just not all there. Okay, it was funny in my head. Okay, so if you're taking a look, again, this is the GI tract. When we take a look overall from the top of that red line all the way down to the anal canal, the very end of that line, that overall we have this four tissue layers that make up that collapsible tube that I'm going to be talking about in a moment. So the innermost layer, the one that's in direct contact with food, is known as the mucosal layer. The one that is just outside of that one is the submucosal layer. Then there is a muscular coat. I told you there's going to be lots of contractions and movement. This muscular coat, or the muscularis, is what's going to cause all of that. And surrounding all of this on the outside is this serosal layer, similar to the types of serosal membranes that I've already spoken to you about. So no matter where we're looking in the GI tract, could be the stomach, or it could be the small intestine, it could be the large intestine, the overall structure of that red line tube that we look at has the same basic structure. There are four layers. Sometimes we have a little bit more, sometimes a little bit less, but overall we have these four basic layers. This mucosal layer on the outside, which is basically epithelial tissue, some connective tissue, and a very thin rim of muscle, and this is in direct contact with food. So food is coming down this way, it's being pushed forward through peristalsis and segmentation, and we'll talk about that a little bit later, and it's also coming in contact with the walls of this tube directly at the level of the mucosa. And just under that is this submucosal layer, which I talked about, and every single portion of that red line has this submucosal layer, and this is the layer that contains lymphatic tissue, blood vessels, as well as some nerves running through it as well, and we'll come back and talk about the submucosal layer in some more detail. For the most part, though, in today's lecture, we're going to be talking about this third layer, which is this muscularis layer. And there are two layers that are normally associated with this. There is an inner circular layer. So when you contract this inner circular layer, what happens is that the overall lumen that's opening in the middle becomes smaller. It contracts the size of the lumen. If you were to contract the outer longitudinal layer, the one at the very top that you see out here, it actually shortens the length of the tube wherever this contraction is happening. So you can imagine one segment is shortening and the next segment has no contractions. And so it kind of looks like a movement of a worm or a slinky moving along. And you can imagine if you can get these moving in order and you can get this outer longitudinal layer to contract in series, you could actually push food forward through this outer longitudinal contraction. And then surrounding all of this on the outside is this serosal layer. You don't have to memorize any more than I've just gone through with you in this particular slide. I'm not going to ask you any details that are sort of hidden up here on the other bullet points. If you're really interested in it, you can go and read that for yourself. Do you have any questions on this before we go on? Yes. Oh, okay. So the ENS, that's a great question. This is the enteric, E-N-T-E-R-I-C, the enteric nervous system. And there's a nervous system that controls how our gut moves. Again, thanks for asking the question. Feel free to interrupt at any time. And as I mentioned or highlighted here, we're going to be talking about this particular layer in a little bit more detail today. And we'll talk about some of these other layers in more detail next week. Okay, so at the very start of the process, and again, you don't have to worry about these slides. I'll tell you the slides that you have to really worry about for your final exam, etc. When we start looking at this particular slide, there's a lot of processing of your food that's going on at the very outset. So your oral cavity, the teeth, the tongue, the palate, the examination of food, it's actually more important than you give it credit for. If you were, for example, to start chewing, not chew your food and to start swallowing it immediately, it could actually be disastrous for your digestive tract, because without you determining whether or not there's something in the food that can actually cause damage to your GI tract, like a bone, for example, or something sharp that could potentially tear the GI tract, would actually not be beneficial for you. So it sounds really trivial, but all of these different processes are designed to help you do a food inspection to begin with. And again, this mechanical processing of food, breaking it up into smaller bits, is a common theme throughout this entire digestive tract system. And in the oral cavity, we have this salivary gland, and these salivary glands will begin this process of chemical digestion that we will talk about a little bit later. Most of you took high school chemistry or biology, right? Yes? Do you guys ever do that experiment where your biology teacher asked you to chew soda crackers or those crackers and keep them in your mouth? You guys don't do those experiments anymore? The good old days. So if you were to lie to me and say that you did do these experiments, one of the things that we were taught in school, which turned out to be really wrong, but we were taught in school that if you're chewing soda crackers, the kind of crackers that you normally get with your soup, if you were to chew those, because they contain lots of carbohydrates, and you just kept them in your mouth for a very, very long period of time. I don't know anyone who would actually do this. And your teacher was to ask you, so how do those soda crackers taste now? You're supposed to say they taste sweeter because they're being converted from carbohydrate into basic glucose. And it's supposed to taste sweeter. Of course, it never really did, to be honest. We all said that because we just wanted to get the experiment over with. But this is sort of where the chemical digestion of carbohydrates begins. And this is where it starts at the level of your mouth with the salivary glands. And we'll come back to that in some more detail. And again, this is sort of the pharynx, the part where we actually use the tongue to push this bolus of food, this the mix of saliva, partially mechanically digested food to the back of the mouth, and then it passes down past the pharynx. And once it's past this pharyngeal phase, and it then works its way down into the esophagus, that's the last voluntary act that you have, right? So how are you making out with that yogurt?
Great.
Are you finished?
I finished.
Okay.
Thank you so much.
Can I ask you for a favor? Can you stop digesting that food now?
I'll try.
You'll try. And that's an honest answer. That's the best that he can do. The only way that he could have really stopped would be up at this phase here. The last control phase that you actually have in your digestive process is when you swallow. After swallowing, when you swallow and it goes into your esophagus, the rest of it is really just beyond your capacity to speed up, slow down, stop, do anything to it, because it's all under autonomic control that we'll come back to a little bit later on. Do you have any questions about this before we move on? Yes.
Just as a curiosity.
Yeah.
I don't know if there was a recent study done in terms of answer this or whatever.
Yep.
Does the uvula play any part in terms of a protective mechanism?
Yeah, I think a lot of these structures, like the uvula, have been shown to have some basic protective functions, especially when you're talking about infections through bacteria, etc. But overall, you'll see that our GI tract system is well designed to actually handle a lot of the different types of microbes and microorganisms that we encounter. Okay, but it's a great question. So again, we're not going to talk about this in a lot of detail. The main thing that I wanted to talk to you about here, this same structure, this esophagus, is really the same structure that we looked at earlier. It's those 4 different layers. It's that muscular layer that allows it to kind of contract behind this bolus of food, and it pushes that bolus of food down toward your stomach. The other thing that I wanted to highlight in this particular slide that we're going to come back to, and is also a common theme, when we look at digestion, we kind of look at this sort of as an open tube. And it is. It really is an open tube. However, between different compartments of this tube, we'll actually find other rings of smooth muscle that actually act as sort of temporary doors or barriers. And these sphincters are going to be really important throughout the remaining lectures that we go through. So for example, when you push food down, it will open up this sphincter down here. And if this sphincter, for example, is not working well, you'll have that acid reflux that maybe some of your parents have after eating. And especially as you get older, some of these sphincters, in this case the lower esophageal sphincter that's between the esophagus and the stomach, the muscle, just like any other muscle, tends to get a little bit weaker, and this lower esophageal sphincter actually doesn't close as well as it should. You eat something that has a little bit too much acid stimulation here, and it pushes its way back up. And because this is all epithelial tissue, it feels like it burns as the acid works its way back up here. There's another sphincter, as you see down here as well, and this is the pyloric sphincter. So we have different sphincters that control the passage of food from one part of this tube to another. Okay, any questions about this before we go on? And again, you don't have to bother memorizing. I'm hoping that you're trying to learn this as we go along rather than trying to memorize this as we go along. Any questions? Comments? Chocolate? No chocolate. Okay, we'll talk about chocolate in a moment. Okay, so I told you earlier on that when we look at the different sections of the GI tract system, that overall it's sort of the same overall theme or topology, the same overall 4 layers that we talked about. And if you look at the stomach, which is still part of the GI tract system, we certainly do have the different parts of the different layers that we talked about earlier. We still have that mucosal layer, we still have that submucosal layer, we still have that muscularis layer. We don't really see it that well here, but we would still have the serosal layer on the outside, so we still have those 4 different layers. But when we look at the stomach, we would also see that there are some unique features that we only find here in the stomach. We don't see this in the other parts of the GI tract system. And the stomach is really the GI tract system that has a particular purpose. And one of the purposes is to actually store food. So your stomach processes food, but it's also there to store ingested food, especially in this upper portion known as the fundus, right above the body of the stomach, where it will actually store food for short periods of time. In the stomach, of course, is where acids as well as other enzymes are going to be secreted to initiate the chemical breakdown of food. But not all of your food that you ingest is actually going to be broken down chemically at the level of the stomach. Let me just see. Okay, so if you're taking a look, this is the muscularis layer, the same layer that we saw before. The stomach does a lot more mechanical processing, mechanically breaking down food, mechanically mixing food and the digestive juices or enzymes together with the food that you've just swallowed. And as a result of that, we don't only have that inner circular layer, and we don't only have the outer longitudinal layer. In between them, we also have this other layer, which is known as the oblique muscle layer. We need that extra muscle, in other words, to cause more contractions and more forceful contractions at the level of the stomach. And again, this is sort of the overall structures that we talked about, the inner oblique, the middle circular layer. And again, this is an important point as well for you if you're thinking a little bit about this, and it might be something that you might want to circle and come back to over the weekend. When we talk about the middle circular layer, this actually helps to form those sphincters that we talked about earlier on. So it helps to control how food gets into and out of the stomach. The lower esophageal sphincter on one end and the pyloric sphincter on the other end, just before it gets into the intestine. And then, of course, the outer longitudinal layer. Do you have any questions about this before I go on? Or anything I can answer for you? Okay. I'm going to tell you that I'm going to cover this slide several times in several different ways. I am also going to tell you ahead of time. So slides 23 coming up, as well as slide 24, are really important slides because there's a lot of enzymatic digestion going on. And this is actually the same slide repeated on slide 24, just shown in a different way. So I'm going to start off with this slide, and then I'm going to take you into slide 24, which just shows you some more details about different cells that we would have. So here are those 4 layers that we just took a look at. And those 4 different layers that we have in the stomach, and this is found only in the stomach, that mucosal layer tends to be a little bit thickened. Okay, so it's a little thicker than we would see elsewhere. In fact, if you take a look, this overall structure, the inner portion, the mucosal layer, actually has these invaginations. It kind of looks a lot like these folds and these ridges that are found, and these rugae are unique to the stomach as well. So if we're to take a look at a more smaller level, one of the things that we would see is in this mucosal layer, and we won't bother about the 2 other layers that we normally see, the 3 other layers, the submucosal, muscularis, and serosal. If we were to take a look at these, we would see that there are some unique cells that we would find in the mucosal layer only at the level of the stomach. We don't see this in the intestine. We don't see this in the large nor the small intestine. We don't see this in any of the other accessory organs. We only see these cell types here in the mucosal layer in the stomach. And they have very, very specific functions here in the stomach. So many of these different cells, including the mucous neck cells, the chief cells, and parietal cells, as well as interspersed endocrine cells that are found within these different gastric glands as part of these gastric pits, so these invaginations throughout the mucosal layer. Each of these gastric pits contains a set of cells which is called a gastric gland, and these sets of cells include things like chief cells, and these chief cells will secrete an enzyme precursor known as pepsinogen. Another cell type that is found in these gastric glands are these parietal cells, which secrete hydrochloric acid. It's not the only thing they secrete, by the way, which is also on the next slide that we'll get to in a moment. So if you're to take a look, these cells are kind of unique. They have specific functions. They all secrete specific things. And I'll just mention one other thing at this point. We're not going to talk a lot about absorption today. We will on in next Tuesday's class, there isn't a lot of things that get absorbed directly from the stomach. Do you know anything that does get absorbed directly from the stomach? The stomach is really there to digest chemically some parts of your food. Not all, but some parts of your food. Do you know what gets absorbed directly from your stomach? Everything else has to get to the level of the intestine. It takes time, in other words. What gets absorbed directly from your stomach? Anything that you're aware of? So some drugs, for example, get absorbed directly from your stomach. So aspirin gets absorbed directly from your stomach. And because you didn't know this answer, I'm assuming it's because most of you aren't drinkers. So alcohol gets absorbed directly from your stomach as well and works its way into your circulation. So this is the same slide shown in a slightly different way. This is sort of that gastric pit. These are the gastric glands that make up parts of the gastric pit, and there are many more functions of different cells that are shown for you. For the time being, for today's lecture, for your weekend homework as you get ready for your final exam, there are a couple of things that I want you to make sure that you're aware of. So the mucous neck cells that you see up here, the name actually suggests what they make. They make mucus, which acts as a sort of protective lining. It's not really that effective, to be honest. When you think about food and you go back to your high school biology class, you kind of hear about mucus protecting your stomach from digestion through hydrochloric acid. That's not true. So what really happens is the mucus and hydrochloric acid come together, and when it comes in contact with food, the mucus actually helps the hydrochloric acid to stick to food better to help it to digest. So it's not just a protective function. It has other functions associated with it as well. The thing that I don't want you to forget about is that the mucous neck cell actually also secretes bicarbonate. So you'll see the reason why we need bicarbonate, because right under it are those parietal cells. On that previous diagram, I showed you that parietal cells secrete hydrochloric acid. So this bicarbonate is there to help to neutralize some of the acidity related to this hydrochloric acid. In addition, these parietal cells also secrete another substance known as intrinsic factor. And without intrinsic factor, you wouldn't actually have very good bones. Your calcium absorption rates would fall, and we'll talk about this in the absorption lecture next week. We have another cell type, which I didn't talk about in the previous diagram, enterochromaffin cells, and this actually secretes histamine, which helps to stimulate gastric acid secretion. The last cell type that I'm going to talk about today, we'll come back to these other ones a little bit later on, are these chief cells, and these chief cells are important because they secrete pepsinogen as well as gastric lipase. Okay, so there are some more things on this slide than there were on the previous slide. So I see one question there and another question from your colleague. Yes?
So histamine stimulates acid production. If you take antihistamines, does that mean your stomach will not produce acid as effectively as it would?
That's correct. Yep. So if you take an antihistamine, your digestion will get affected. You are correct. Did you have another question?
So is calcium absorbed through the stomach as well?
It is not.
No.
No. You'll learn the mechanism by which intrinsic factor actually causes calcium absorption at the level of the small intestine next week. Okay. Any other questions that I can answer for you now? So this point here about gastric lipase, this has actually been somewhat controversial for a very long period of time. And it turns out that over the past few years, we used to think that the pancreas was really the only place that really secreted lipase, but more and more investigators have shown that lipase can get secreted by your stomach and by lingual sources as well. So in your mouth as well as in your stomach. And we'll talk a little bit more about that later. And lipases are there to help in the digestion of fat. So it breaks fat down into short and long chain triglycerides, the first breakdown products, as well as free fatty acids. But if you were to take a look at all of this together, and if you know a little bit about nomenclature and how we name things, do you know what of the three different food products that we eat, carbohydrates, fats, as well as proteins. Which of these do you think the stomach is really good at breaking down? Is it good at carbohydrates? Lipids? Okay, so the lipase says it's good at breaking down lipids. I would agree. Anything else that it's good at? Protein? Okay, why do you say protein? Because of hydrochloric acid? Why protein and hydrochloric acid? Okay, so generally speaking, we have proteins which then can be broken down into polypeptides and then into peptides. So if you're thinking about the word peptide, do you see anything that resembles peptide up here on any of these different cells? So this chief cell, this pepsinogen, is one of those factors that helps in the digestion of proteins by breaking down proteins. It's a peptide bond breaker. So it breaks down bonds that are found in larger molecules like proteins. So we'll come back and talk about these later. I do want you to know the different cell types and the different types of secretions that they all have. I don't believe in being disingenuous, so these are questions that I have submitted, and I think that any physiology student should know, at least remember for short periods of time, the different types of cells that are in the stomach that aid in different types of digestion. So this is, again, another slide that I want you to be familiar with. I want you to think a little bit about this before we take a break. I'll try to get through about 2 more slides and then we'll take a break. I'll ask for some more volunteers. Hopefully I'll get a few more volunteers this time around. If you're to take a look at those gastric pits that I just outlined for you, and I'm going to tell you that the gastric acid, the hydrochloric acid that gets mixed in with some of that mucus, even though it's a protective lining, it's not a very effective protective lining. That mucosal lining that we see, that whole surface layer, so we have mucosal, submucosal, etc. If you're to take a look, because of the sheer amount of acid that gets produced, because of the shearing forces, remember you have 3 muscle layers underneath, and then you have all this acid being dumped on top of these mucosal cells. So it's a hostile environment, as I mentioned to you earlier. It's not just hostile to bacteria, it's hostile to you. It's hostile to the mucosal cells of your own body. So if you're to take a look at that, the mucosal lining of the stomach actually has to be replaced every 3 days. You have a brand new stomach, in other words, the mucosal part of it at least, every 3 days. If you're thinking about that, again, in some more detail, and I won't ask you about the calculations, and nor do you have to worry about the calculations here. I'll tell you which parts that I want you to actually think a little bit more about. Every single minute that you're sitting here, you're losing about 500,000 cells. About 500,000 of your cells are gone every single minute. You're not eating food or anything else. It's just the natural part of your stomach being a hostile environment that is causing the loss of a tremendous number of your cells. Now imagine they're not being replaced. That would be a terrible thing. And so we know that there have to be stem cells inside your stomach that actually allow for the replacement of all those cells that you're losing every single minute that you're just sitting here doing absolutely nothing. And this discovery of these LGR5, and LGR5 is a protein that we find and we can detect in these stem cells that we find at the base of these gastric glands that are part of the mucosa. These are the ones that will eventually climb up to the isthmus, and from the isthmus will form different progenitors which will form every last one of the cells that I've shown you in here. And that's the function of a stem cell. It's because we're losing so many of these. So what do I want you to take away from this? Well, I don't need you to memorize the numbers because that's not important, to be quite honest. You're going to remember those numbers anyways, but the fact that we're losing so many of these cells, we need to have stem cells. And one of the ways that we mark these stem cells is through this protein LGR5. And so seeing something that is LGR5 positive means that it is a stem cell. And these stem cells normally reside here at the base. And then as they start forming into these isthmus cells and they move up toward the isthmus, they can then form every other cell type that we find within these gastric glands. And this is how we reproduce these cells. If we were to suddenly lose somehow all of these LGR5 stem cells, your stomach would be gone within that 3-day period. So we know that these stem cells are incredibly important for us, and that they're really important in maintaining the integrity of the mucosal system. So I'm just going to go through this one slide, then we'll take a break if you don't mind, and then we'll come back and try to finish off today's lecture. So this small intestine, you'll often see this referred to as the small bowel, and it's because of the size of the lumen. Overall, it's really quite long. It's about 7 meters in length. And again, this question here, can you think of a reason why you need about 7 meters of length of intestines? Why do you need such a long distance? Any ideas? Yes, at the back. So to increase the surface area, to increase the length of time that food is in contact with it, to increase the overall effectiveness with which we're absorbing food. You've gone to great lengths to eat that food. In order for you to actually make use of that food, you actually have to absorb it. In order to help you absorb it, this small intestine has to be as long as it possibly can be. And your colleague is exactly right, to also increase the surface area for that absorption to occur. So it occurs right after the emptying from the stomach at that pyloric sphincter, and then it ends at another sphincter. Here we call this a valve at the level of the ileocecal valve, the junction between the small intestine and the large intestine. And this is really important. This is where we're going to talk a lot about motility, how these muscle contractions, those two layers, the longitudinal and the intercircular layer, work together to push food along, as well as to cause segmental mixing of food. And because of the different functions, that 7 meters is not all doing the same thing. They're all useful for absorption, but in fact the duodenum, the shortest part of it, and the longest part, the greatest part for the absorption, the jejunum and the ileum, all have slightly different functions that we'll talk about after the break. Okay, so I think you usually take a 10-minute break at this point, no? Yep, thank you. Okay, so let's take a break now, and then we'll come back and talk a little bit more about this.
Mr. J, just for the exam, I wanted to know, is Physio X also here or not? I just saw that it was TA to 10. Oh,
I submitted 12 questions for this section.
okay, all right.
6 from this first 2-hour block, and then 6 from the, I have no idea.
I'll email.
Ask Dr. Wong.
Thank you.
Hello.
Quick question.
Yep.
Wouldn't it be also efficient in digesting carbohydrates, just sheer mechanical, like, absorption of the breakdown of
You know, the
food?
bolus of food that enters into the stomach after mixing is really big,
Oh, really?
size.
Oh, okay. So what if somebody, like, you know, like children sometimes like a bit of salt, like that doesn't get breakdown even further?
No,
Oh, that's really interesting.
it's
Thank
why
you.
your mom tells you to chew food.
Yeah.
You actually can't absorb food unless you actually mechanically break it down and then chemically break it down.
I just have a clarification question. When you were talking about swelling, you said that up until the preliminary stage is use control, or after the preliminary stage?
Up to, and then once it enters into the esophagus, and the esophagus, the lower part of the esophagus is all smooth muscle, there's no skeletal muscle, there's no mixed muscle, then it's under your control.
Okay, so because I'm interested in going to speech pathology, so like through volunteering, I've seen this like swelling studies. They've done like they teach patients how to help themselves swallow through that preliminary phase. So I always thought that that wasn't an area that we could normally control.
No, I think you can control the preliminary phase.
Okay.
That's okay.
But so if someone gets a when you exercise too much and then a part of it's not. It's like a rip in the leg. Ulceration. Yeah. Hernia. Is that a ripping of the
They'll push through the muscle as well, but no, that's where actually has to true muscle, not GI tract system, but the muscle that controls the abdominal muscles.
okay, so also, how would you be speaking to one of your students? Yeah, he said, I'll consider taking HB202 this summer, and I'm trying to get into nursing school. I'm wondering if it's a feasible course.
I don't know how useful it is for it's basically in undergrad GP training.
Okay. Are you teaching at this time?
And no,
Question?
yeah, of course.
Big question. Yeah. Or like where is it digested or is it not digested?
And get digested throughout the entire epithelial lining. We always have dendritic cells, part of the immune response, and reactive cells. And so you will have inflammation. If you're allergic to stuff here, you're allergic to stuff inside. So it's unlikely you're going to get a lot of food. My guess is if you really want to food, I would probably not be able to either relieve renal or body. Really, really setting off the immune system.
But then I have been to so is that reaction?
It can be a lot of things. Talking a little bit
Right.
but those are good questions. It's just like something you want to learn. Just causing your the problem is what's my first is they all Okay, I don't know if it's okay with you, but if you don't mind, I'm going to just try to go on and try to cover as many of the slides as I can before we get to the end of the natural break. If we don't make it today, then I'll try to make up for the remaining slides in the next lecture. So as we look at the small intestine, one of the structures that I said started off the small intestine, that 7-meter long absorptive structure, is this duodenum. And it's the thickest as well as the shortest of the 3 different sections of the small intestine that we talked about a little bit earlier. And so it actually makes this little C shape. It actually wraps around the head of the pancreas. So the pancreas itself can be split into the head, the body, as well as the tail. And we'll see that in the next slide. And some of this makes sense in the particular case of the duodenum, because both of these different structures that you see here, the gallbladder and the pancreas, release their substances directly into the duodenum. And I told you that all of these have different functions. So something is unique about the gallbladder as well as the liver, and it will release bile into the duodenum, and the pancreas will also release its secretions into the duodenum as well. And those get into those accessory organs that I talked to you about earlier on. So this is where we get a lot of secretions going into the small intestine. The next section, just anatomically, because we'll come back to this functionally in a little bit, the next bit of this is the jejunum and then the ileum. And it's not like you suddenly go from here is the duodenum and here is the jejunum. It's more of an anatomical term rather than there's a really distinct demarcation. And the jejunum is just a continuation. One of the things that makes it really noticeable is that there is a decrease in the diameter. It gets smaller and smaller. It gets harder to push food through because the diameter, that lumen that we saw earlier, is gradually decreasing in diameter as well as in the thickness of the wall. So the tube itself, the lumen, is getting smaller, and then the outside part of the tube is getting thinner and thinner. All of this suggests that we're slowing food down, increasing the amount of absorption, and then making it easier to absorb food across this thinner lining, or the thinner wall thickness that we see going on here at the level of the jejunum. The jejunum, as I mentioned, is really designed to help you absorb it as much as possible. And as we exit from the ileum, the last part of the small intestine, before we get into the colon, we run into this ileocecal valve, or IC valve, and that's ileocecal, between the ileum and the cecum, or the colon. And this ileocecal valve is important for a couple of reasons. All valves, or sphincters, control the movement of food from one section of the digestive tract into another. And so if we have this open or closed, it actually will change the rate in which food is moving from one section to another. So the ileocecal valve helps to prolong the amount of time that food stays in the small intestine. So it helps to increase the overall absorptive time. The longer it's in the small intestine, the more you're going to absorb from it. And then equally important, anything that's undigested, because most of the food that works its way to the ileocecal valve is undigestible material. Everything that you wanted to take away from it you have, and everything that you can't take away from it, it gets pushed beyond this ileocecal valve. That also prevents this backflow of any undigested material that we store in the large intestine until we have to expel it. Okay, so this gets into this whole issue of motility. Throughout this entire length of the small intestine, anywhere actually within the GI tract, but particularly within the 7-meter length of the small intestine, this food has to be moved along. It can't just, it's not just kind of flowing down by itself. It has to be assisted by these different muscle contractions. And we've already talked a little bit about these muscle contractions. So built into the walls, and we'll come back to this in more detail later. And again, you don't have to bother trying to get down all the details now. I'd rather you try to pay attention to the big picture, understand what's going on, and then I'll tell you the sections that you need to maybe worry about for your final exam. But if you're taking a look at the small intestine and the smooth muscle cells, and you guys have had lectures on smooth muscle cells, right? So you know that smooth muscle cells, some of them have pacemaker activity. They have like rhythmic contractility, rhythmic depolarization, and these pacemaker cells are also found at the level of the small intestine, and they help it to spontaneously contract. And we'll talk a little bit about this in today's lecture. And all of this helps in what's known as peristalsis. It helps this bolus of food that is coming into the small intestine to be pushed forward. And it's this series of contractions between the longitudinal and the circular muscles that allow this bolus of food to move forward. And this is one of the reasons why you can eat upside down. It's independent of gravity. It doesn't need gravity for this movement of food forward. And it's really an important function that the small intestine actually has. Okay, so in terms of the last part of the overall line 1, if you want, or the red line that we talked about earlier on, this is the large intestine or the large bowel or the colon. It's relatively short. It's not that 7-meter length. It's at 1.5 meter length. And again, it starts at the ileum and then it ends at the anal canal. And overall, it really doesn't serve much more of a function as far as we can tell, other than causing the absorption of some water and electrolytes. So there is still water, electrolytes left in that undigested material. It can get absorbed at the level of the large intestine. It's also there to store waste. And also, most importantly, nowadays, when we talk about the microbiota and how bacteria colonize your body, especially your GI tract, this is where it colonizes the most. You have the greatest concentration of bacteria here at the level of the colon. Do you have any questions about this or the previous slide before I go on? No? Okay, so if you were to take a look at the overall structure, this is the colon. So this is the ileum. So the small intestine would be all up here. Then it works its way into the ileum. Then we get into the large intestine and then into the anal canal. One of the things that we would see as we get into the large intestine is this blind-ended sac. So it's this structure here, and this is what's known as a blind-ended sac or a blind-ended pouch. And this structure has associated with it this little vermiform appendix, and it's right after this ileocecal valve. And one of the interesting things about this vermiform appendix is that we have a lot of different lymph nodules here. And have you guys had a section on immunity already? So you know the function of the lymph nodes and the maturation of your immune system. So I told you earlier on that your GI tract system is in contact with a lot of different bacteria and foreign microbes, and it needs a really strong immune system built into it. And throughout the entire length of the GI tract system, we have a number of immune cells that are found, and we also have these different lymph nodules. We're not so sure if the lymph nodules that we find in the vermiform appendix are really that important for immunity. We do know that immunity is really important overall in terms of the GI tract, but we still don't know the exact function of this vermiform appendix. And again, just out of curiosity, how many of you have had an appendectomy? Like you've had your appendix removed because it was inflamed? So a couple, just two. That's actually statistically that's about right. So young individuals typically don't have any issues with their appendix. As you get a little bit older, you might run into some issues with the appendix and have to have it removed. Sorry, you had a question?
Yeah, in terms of pH between the first part of the intestine,
Yep.
is there a drastic change because there is more secretion in the first part?
So your colleague was asking between the first part of the small intestine and the last part of the small intestine, so from the jejunum or duodenum all the way down to the ileum, is there a change in pH? There is. We're going to talk a little bit more about it, but it's probably not as dramatic as you're thinking it is. Okay, and we'll talk about some of those secretions in just a few minutes. Okay, so some of those accessory organs, to go back and finish off, we just looked at the overall structure of line 1. We looked at the sort of gross structure of how it has 4 different layers, that in some parts of the GI tract system that it has some modifications. Sometimes we have 3 layers of muscle, sometimes we have different mucosal cells that are associated with it. And then we also talked about the fact that feeding into line 1, we also have these accessory organs as well. And we'll start off with some of them, talk about the salivary glands, and then work our way down into the pancreas. And hopefully we'll be able to answer some of the questions that your colleague had earlier. So again, I'm not going to lie to you. I did submit 12 questions earlier to your course coordinator, and there are 6 from today's lecture at the very beginning, and then 6 from next week's section. And one of those 6 questions from today's lecture comes from this particular slide. And there are a lot of different things on this particular slide that I want to talk to you about, some more important than others. The salivary glands that we have, I mentioned at the outset, is really involved or thought to be involved in the digestion of food. We don't really tend to, especially in today's society, hold food in our mouths for very long. Like you literally shovel food into your mouth, and if you've ever come to my home and ate around our table and you happen to see my 17-year-old, you would be surprised if she actually took more than 2 bites, because it looks like she's literally wolfing the food down and not even bothering to chew. So we don't really spend a lot of time digesting in the mouth. However, when we take a look, the salivary glands actually have a number of different functions, and we have a number of different salivary glands. The parotid salivary glands up here at the top, the ones that are just found under the mandible, the submandibular glands, ones found under the tongue or the sublingual salivary glands, etc. And all of these are thought to not just provide lubrication, but also to provide the digestion, the beginnings of the digestion of complex carbohydrates. So if you were to eat things that have carbohydrates, in theory, the salivary glands, because of what they secrete, will actually begin the digestion of carbohydrates chemically through an enzyme that is found in the secretions known as salivary amylase. In theory, we have another lipase, lingual lipase, that in theory begins the digestion, the chemical digestion or the chemical breakdown of fat called lingual lipase. There's another factor that is produced by the salivary glands that we take for granted, which is known as epidermal growth factor. It is an important growth factor for a number of different tissues, and we produce a lot of this over the course of a day. So if you're producing about 1 liter to 1.5 liters of saliva every day without engaging in too much eating or excessive eating, just on an average day, it's about 1 liter to 1.5 liters. We're producing a lot of this epidermal growth factor from these salivary glands as well. The vast majority of this comes from those submandibular glands, about 25% from the parotids, and very few or a very small amount of these secretions come from those sublingual glands. And again, you don't have to worry about the histology that I normally talk about to other classes related to these different salivary glands and these serous cells of these salivary glands. Do you have any questions on this before we go on? As I mentioned, this is a slide I'd like you to spend some time this weekend, at least going back and reviewing. And that's probably the best way, and what I would suggest that you do. The slides that I've mentioned, go back, review them, see if there are any concepts that you're not comfortable with or things that you're not clear about, and then come back and talk to me next Tuesday. Do you have a question? So we think that EGF has a number of different functions. Some of that might be protective along the GI tract. It is a growth factor. It might be restorative in some ways. We're not 100% sure. We do know that there's a lot being produced. I think there is a question at the back as well. Yes? So the ducts actually just convey, so the glands produce these different molecules and enzymes, and then the ducts actually convey them into the mouth in order for them to mix together with the food that you're processing. Okay. Okay, so and we'll actually come back to the actual salivary amylase a little bit later on. So another one of those accessory organs that feeds into line 1 is this liver. It's the largest gland in the body. And again, it's for your own information. I'm not going to be asking you about this. It's actually a really important organ, and it's right under that diaphragm that helps you to breathe. Generally consists of those 4 different lobes. And I think that you've had lectures on the liver before and the hepatic system. Yes? So it has a number of different metabolic functions. It's really important in blood formation and removal of damaged blood cells, etc. And along the way, it also produces the substance that in the digestive system that we're the most interested in, which is bile. And all of these different hepatocytes in these 4 different lobes are there to produce that bile. And there, again, you can see that falciform ligament that we talked about that helps to make sure that the liver is in the correct orientation and in the correct spatial location. So again, this is a really busy slide, and I don't need you to memorize all of this slide. There are a couple of things that I wanted to point out for you, just to orient you a little bit for your final exam. Your liver has a number of specialized cells. There are lots of specialized cells, but within the liver, these Kupffer cells are really important for phagocytosing and antigen presentation of things brought in to your liver. Of course, you've already talked about these other factors like plasma protein production, like albumins, with other instructors in the course, so we won't go into that. And again, just in terms of digestion, the liver is where we store for short periods of time glucose that we take in. It's not a long-term storage solution for glucose. We keep it around for short periods of time in the liver within hepatocytes. Again, it has a number of clearing functions, removing antibodies and other proteins, removing toxins. And again, we won't worry about all of those because I'm sure you've covered those in other portions of the course. For us, again, we're more concerned with this function of bile. So these Kupffer cells is one aspect I'd like you to make sure you know. And this bile is another function that I want you to be aware of. And it contains a lot of water, cholesterol, fats, these bile salts, some proteins that we won't get into details about, as well as this breakdown product known as bilirubin. But overall, we just call it bile. And one of the things that this bile does, and you don't have to worry about the anatomy of this biliary system. There are a number of different duct systems that connect the bile that's produced in the liver to other parts of the digestive system. But ultimately, wherever they're going, whether they're going through the cystic duct and into the gallbladder, etc., all of them empty out into the common bile duct. And this common bile duct all empty into this duodenum that we talked about earlier. And so this is this biliary system where we have the hepatocytes forming bile, sending bile to other parts of the biliary system, including the gallbladder, which concentrates the bile, and then all of it gets emptied out into the duodenum, and this is the main focus of this particular slide. Don't bother trying to memorize all of the different order and the order of operations of the different parts of the biliary system. I think you have a modified version of this slide, if I'm not mistaken, right? Okay. Do you guys know where your gallbladder is? Do you guys all have a gallbladder? Is it a trick question? I don't know. I mean, it's either yes or no. Do you have a gallbladder? As far as you know, yes. Okay, good. Can you live without a gallbladder? Yes? You say that with real confidence. Not sure now. I've shaken her confidence. You can indeed live without a gallbladder. So one of the functions of the gallbladder, and some of you will know that you can get gallstones, and these are these sorts of gallstones that you can see on an ultrasound. If you were to excise someone's gallbladder, and this is a serious case of someone who ate a lot of fatty food. If you eat a lot of fatty food, you're more likely and more prone to develop gallstones. So again, bile, which is produced by the liver, is actually there to help you digest fat. So it's counterintuitive that eating more fat actually produces these gallstones because it's trying to actually help you in that fat digestion. And it is actually a really painful condition. These gallstones, if they grow big enough, actually prevent things leaving through those ducts. And every time this gallbladder contracts and tries to squeeze out that bile, it hurts. And especially after you've eaten a fatty meal and the gallbladder has these gallstones and it contracts and tries to push out that bile, it can become very, very painful, which is one of those signs that people go in to get these ultrasounds, see this on an ultrasound scan, and then ultimately have the gallbladder removed. It's not a very large organ. It only fills up to about 40 to 70 mils, depending on your size, your sex, etc., and your age, in terms of the amount of bile that it can actually hold. It's a little different. It's not the main line that we see within the digestive system. We don't have those 4 layers. We see many of the similar layers, and serous outer layer, a muscular layer. It has to contract, in other words, to push that bile out into a duct, and then this mucous membrane. But ultimately, it's really there to store bile for the times that we need it to help us with fat digestion. So it stores bile as well as concentrates bile to make it more effective. And if you have too much cholesterol, it actually causes the production of these different gallstones. So this gets into one of the other accessory organs, which is the pancreas. And the pancreas actually is, in many ways, the most valuable sort of accessory organ that you have. All these other ones are really important. But I just told you that you can live without a gallbladder. You don't need it to digest fat. Your liver can make up for some of that. And you can restrict your diet. You can live a long, healthy life without a gallbladder. You can't live without a pancreas. If your pancreas is gone, you can't survive. And hopefully by the end of this lecture series, you'll understand why this pancreas is actually so incredibly important. So you've talked about the pancreas in a different context. You've talked about it in the context of the endocrine system, right? So you know all the different types of hormones that are released by the pancreas? Can you name them all? You could if it was on a multiple choice test question sitting right in front of you and someone had a Scantron right beside it, right? But if you were to think of the pancreas sort of in a different way, so if we were to take a look at the structure of the exocrine pancreas, which releases into a duct, which then releases into the duodenum, which we'll talk again about in a moment, you'll see that this exocrine pancreas has a number of structures that are associated with it that you see right here. So each of these represents parts of the exocrine pancreas, and you'll see that there are different ductal cells that are associated with it, and you'll see that they secrete different factors like bicarbonate and other things. And you'll see that this overall structure from the head all the way to the tail allows you to be able to secrete lots of different things into this duct system, which eventually empties out into the duodenum. So some of these secrete things like bicarbonate. The acinar cells that you see here in yellow, they will secrete these digestive enzymes that I'll show you in a few moments, and these are going to help in different forms of digestion. We're not going to talk about these islets of Langerhans that you've looked at that make up the alpha beta cells that secrete insulin as well as glucagon. So we're looking at these the exocrine portion of the pancreas, and these digestive enzymes that are secreted by the acinar cells, that get released into the duodenum are really, really important for us. We can't survive without these. We can't digest food efficiently without these. For example, we talked about in the stomach, the digestion of proteins, a little bit of lipids, and we've talked about carbohydrates and salivary amylases, etc. But it's really the pancreas. The pancreas secretes lipase. The pancreas secretes different peptidases that help in the breakdown of proteins, and it's also involved in many of the breakdown products for carbohydrates. We can't survive well without this exocrine pancreas. And we won't talk at all about the endocrine regions of the pancreas in this lecture series. So I'm going to switch gears just a little bit for the next few slides and talk about some of the modifications. It's for your own information. You don't have to bother trying to memorize them. Some of them we've talked about already, and some of these modifications that occur are useful for us to have as a frame of reference. So, for example, the surface area in the stomach that we've already talked about get increased through those rugae, those folds that we saw at the mucosal level from that earlier slides. And the increase in that surface area is also increased by those gastric pits that we talked about, those gastric pits with those gastric glands, with those cells that I need you to memorize for whenever your final exam is. And that at the very base of those gastric pits, where we talk about those LGR5 positive cells, those stem cells, I need you to make sure that you are going to be comfortable with that on the final exam as well. But this is where we increase the surface area. And we know that these other modifications also increase the surface area throughout the entire GI tract system for different reasons. We want to increase the surface area. In the stomach, we increase the surface area in order to increase chemical digestion of food. In places like the small intestine, and I have to apologize for this rather small abbreviation SI as small intestine, there are a number of different modifications that you don't have to memorize. It's for your own information. Look at the number of different modifications that the small intestine has. One, we already know it's really, really long. We also know the lumen size changes. We also know that the thickness of the wall for absorption also decreases. But in addition to all of those things, being 7 meters long, etc., the small intestine is designed to increase surface area to absorb things as much as possible. So we decrease the transit time, we keep things in there longer, we make the tube itself longer. But in addition to that, we have structural changes like these Keck ring folds or circular folds, and these are these permanent folds that you see that increase the surface area. Then on top of that, increase in surface area, we also have these mucosa villi, and these villi are these finger-like projections that stick out. And on top of the villi themselves, if we were to take a microscopic look at the villi or finger-like projections, we would also see microvilli. All of these different factors increase the surface area within the small intestine 30-fold. So when we're talking about the stomach, we're talking about having an increase in surface area for chemical digestion. Here, this is designed as much as possible for absorption, and we'll talk about that. And these microvilli, again, increase this further to this 500-fold increase overall from start to finish. You're going to have to remind me, how much anatomy do you get in this course? Enough to make you feel good at the end of the day? So I have to apologize. I thought you guys got a lot of anatomy in this course, and one of the things that I put on your final exam was this, that we also next week have to talk a little bit about how we absorb things into the blood. So in other words, we need to talk a little bit about the blood supply to the different organs that we have just been talking about. And this is where you need to know a little bit of anatomy. So if you don't, this is a slide that perhaps you might want to come back to over the weekend. And again, I'm coming back on Tuesday, so it's a good chance for you to take a look at 6 to 7 slides, make sure you're comfortable with them, and come back and chat with me on Tuesday if you're not comfortable with this. But if we were to take a look at what's known as the splanchnic circulation, which is the blood supply throughout the GI tract system, if we were to take a look at the stomach, the majority of the blood supply to the stomach is through this celiac artery coming off the main aorta. So the stomach is supplied by this celiac artery. Okay, and that's one thing I'd like you to make sure that you're aware of. And the small intestine is supplied by the superior mesenteric artery. And the large intestine, even though it's physically in close proximity, is actually supplied by two different um, arteries, the superior as well as the inferior mesenteric arteries. So that means that there are different blood supplies. The celiac artery is not the same as the mesenteric, nor is the inferior the same as the superior. So the blood supply is a little different regionally for these different structures. And these are the different artery systems associated with the different structures that I'd like you to make sure that you're okay with. Will you be okay with that? Okay, cool. All right. So ultimately, going back to our architecture of the GI tract, and we'll come back to the blood supply in the next lecture on Tuesday. When we look at the overall function of motility, there are a couple of reasons why we want to do this. One, we need to mechanically mix food together with the digestive enzymes that we're secreting. We just been kind of circling around this in different ways, where we've been looking at different structures like the gallbladder, the pancreas, etc., secreting different substances, the stomach secreting different substances, but it actually physically has to be mixed in there with the food. There's no use in having salad dressing sort of just sitting on the top part of your salad. You need to mix it all the way through, and that's part of the reason why you mix your salad. You need to mechanically mix your digestive secretions together with it as well. You also have to allow for the contact with where these things are actually going to be either secreted, the enzymes are going to be secreted, or where they're going to be absorbed. So there's a lot of mechanical movement, as I mentioned. So we're not going to spend a lot of time on these two elements, mastication or chewing, and deglutition or swallowing. And we've already talked very briefly about peristalsis, that forward movement, especially in the small intestine along the GI tract. And then there are other things that we haven't talked about, like segmentation, mixing, as well as intermittent propulsive movements. So again, we'll take a very quick sort of break to make sure that we understand everything. And this is where, if you have any questions that you're not sure of, now's the time to ask. Or if you have any other things that, as someone who's just interested in some of the things that we talked about, now's your chance to ask as well. So we're going to go on and talk about some of the different factors that get secreted in more detail. And then we'll talk about some of the different factors that are part of the stomach and small intestine. But before we do that, do you have any questions or things I can clarify for you now? Yes, hello.
The gallbladder, was that located right next to the pancreas?
It's very close to the pancreas, correct.
Had the same ducted as the pancreas.
Sorry, it has.
They use the same duct to release. Oh no, that's stored the bile.
Okay, you're okay? Okay. Okay, so a couple of things before we go on. Hopefully most of this is somewhat of a review. If it's not, it's okay too. It's just useful to make sure we're all on the same page. We talk a lot about enzymes. We talked about salivary amylase, that's an enzyme. Lingual lipase, that's an enzyme. Gastric lipase, another enzyme. We talk about pepsinogen, which is sort of a precursor to an enzyme that we'll talk about in more detail. All of these different enzymes, every last one of them, and ones that I haven't talked to you about yet, every last one of these enzymes that we talk about act as catalysts to produce a chemical reaction known as hydrolysis. So what is hydrolysis? Any ideas what hydrolysis is? It's a chemical reaction? Yes? Yes, did you have a... Yeah, so what these different enzymes do, and it's really simple. All of these enzymes are there to force a water molecule between a bond in order to break them. So they'll take something big, force a water molecule between it, and break it. In that process, if it's a carbohydrate, it will break it through hydrolysis. If it's a fat, it will break it by introducing a water molecule into triglycerides and free fatty acids. And this is the same process over and over again, known as hydrolysis. Again, a basic principle that hopefully all of you will recall, and if you don't, that's okay too. Enzymes all optimally react at an optimal or specific pH. Okay, so they all work best at a specific pH. And that's going to be important in a few moments. And we'll be talking, I mentioned at the very outset that the pH changes are one of those common things that we see throughout the GI tract system. We start off with a pH in your mouth at about 6.9, and then we get all the way down to a pH of 2 in your stomach, and then it has to go back up to a pH that's fairly neutral at the level of the small intestine. There are a lot of pH changes throughout the GI tract, which means that all of these different enzymes are being turned on, turned off, and being regulated by these changes in pH. There are a lot of stimuli for the different enzymes to be secreted, and some of it is just having food around. That will be enough to cause a secretion in the stomach, for example, or in the small intestine. It might be thinking about food, makes you start secreting. It might be someone who's sitting beside you who brings in these really great smelling poutine, which happens to be my favorite, very aromatic, tastes good, makes you want to salivate, and that whole process is involving the autonomic nervous system, etc. So again, stimulating factors include distension, or simply the movement of the GI tract because of the presence of food, and that will activate receptors that are built into the walls of the GI tract system, and it will generate action potentials causing stimulation. You can also activate the autonomic nervous system, the parasympathetic fibers. If you activate these, you will increase secretions throughout the length of the GI tract system. And you can also use gastrointestinal hormones that can also inhibit or stimulate secretion as well. And we'll talk about this last factor in some more detail next Tuesday. Parasympathetic fibers, which I know you've covered, which parasympathetic nerve do you know the best? Which is your favorite? The vagus. Why is the vagus your favorite? Sorry? Because it connects to the heart? Okay, sure, I'll buy that. The vagus is the dominant sort of parasympathetic innervation everywhere from about here all the way down to here, and guess where your GI tract is? It's right in the middle, and this is why the vagus is so important for us as well. Again, we've talked a little bit about this mechanical digestive process where we chew, and that increases the surface area that allows different chemicals or enzymes to mix, whether it's lipases or amylases. That swallowing and moving the bolus, the mixture of food, saliva, and enzymes to the back of the mouth or the pharynx is important because it also temporarily inhibits breathing so that you don't choke while you're swallowing unless it is completely involuntary. And then we'll talk a little bit about this chemical digestion in the next slide. But this is one of the things that starts this whole process off, as we've mentioned. So again, this goes into some of the different themes that I mentioned from the outset. So if you take a look at the salivary glands, we talked about these submandibular, the sublingual, and then the parotid glands that make up the salivary glands. If you take a look at your saliva, because I know you all take a look at your saliva on a routine basis, right? If you take a look at your saliva, there's actually two different portions to it. So don't do this now because it'd be gross if you did, but if you stick two fingers in your mouth and then you were to get some saliva and then you were to pull that apart, in theory, you would actually be able to see little beads and then little string of water in between. And that's because saliva actually contains a mucus portion as well as a serous or watery portion. Again, serous just meaning fluid. And that's because of the different enzymes that make up saliva. There's also pure mucus only that's part of your secretions that go on in your mouth, and then that serous portion has that serous amylase that we talked about, which begins the digestion of starch. Salivary amylase is an enzyme that breaks down carbohydrates by that process of hydrolysis, breaks things in by inserting a water molecule between carbohydrates. And this begins the digestion of starch. The other thing I told you is that all of these different enzymes work at a maximal or optimal pH. Here, the optimal pH is 6.9. So your mouth, in other words, is slightly acidic. It activates salivary amylase. It's great. Salivary amylase works great up here, but just remember this when we talk about what happens at the level of the stomach. So as long as we have these polysaccharides as part of these starches, and we have salivary amylase at a pH of 6.9, it will introduce water molecules and it will break things down into disaccharides. And disaccharides include things like sucrose, it includes things like galactose, etc. And so in theory, carbohydrates can taste sweet if you leave them long enough in your mouth. Again, some of the things that cause saliva to be stimulated and released include parasympathetic stimulation of those different glands. Just thinking about it activates these parasympathetic fibers, etc. Okay. So when we talk about these different sections, and I was talking to one of your colleagues during the break, I'm not going to ask you to know the details here. We're going to come back to the esophageal response later. This oral response is that chewing, the examination of the food. The pharyngeal response is where you are pushing it back toward the pharynx and then swallowing, and that the uvula is covering those different portions that prevent you from choking. And then we'll talk about the rest of this, which is the esophageal response, a little bit later on. I wanted to talk to you about this sphincter system that we had here at the lower end. So between the stomach and the esophagus, we have a sphincter, as we normally do throughout the GI tract system. And this relaxes to allow entry of food into the stomach and then closes behind it when food gets into the stomach. I told you a little bit about gastroesophageal reflux disease, where this sphincter actually no longer closes well. And especially in older people, you get that acid reflux coming back. And again, we talked a little bit about how things move along this GI tract system because of those smooth muscles. And that's primarily the reason why we can't control digestion. We can't stop. Like even if I really wanted to stop digestion, like I just felt really uncomfortable for whatever reasons, I just had a great dinner and I want to hold on to that dinner as long as I can, and I want to stop the digestive process, I can't, because it's all under the guidance of these smooth muscles, and the movement and contraction of smooth muscles is not under voluntary control at all, although it can be controlled through autonomic regulatory pathways. And this is one of those sort of slides that I'll end off with. I think I have like 2 more slides left, and then we'll break a little bit early today if that's okay. When we take a look at next week's class, and we kind of come back, and hopefully you'll remember most of what I talked about before we get into some of the other aspects. A lot of students have this misconception that, you know, it's really the nervous system and then it's these secretions. Actually, what this diagram is suggesting is that when we take a look at the overall structure, and this is that 4-layer structure we've talked about over and over again, the mucosal cells, the submucosal, and the muscle layers that we talked about in the serosal on the outside, that running throughout this is that nervous system that we haven't talked a lot about. We've mentioned it a few times. And if you take a look at one of the things that it does, is definitely does control the contraction of muscles at different layers. It causes the longitudinal muscles to contract, it causes circular muscles to contract, causes peristalsis, etc. But that same nerve impulse might also be causing the activation of these enterochromaffin cells. And these enterochromaffin cells, as we talked about at the very outset, do you guys remember what substance these enterochromaffin cells release? So remember that slide that I told you that was really important? I think it was slide either 23 or 24 that you should make sure that you know. One of those factors that gets stimulated when a nerve impulse stimulates it is this enterochromaffin cell, and this enterochromaffin cell in turn will release things like histamine, and histamine in turn can stimulate acid production from the parietal cells that are nearby, etc., etc., causing this whole cascade to work. So I don't want you to get into this idea that these are two totally different and totally separate pathways, that the nervous system actually can cause secretions, and we'll talk more about that in the next lecture. So I'm hoping that at the end of today's lecture, there are going to be a couple more slides just to end things off, but I want you to be thinking a little bit about the mechanical and chemical events that occur, some of the different cell types that we've talked about, and then in the next lecture, we're going into more detail. We're going to build on what we talked about here today and get into more of the rest of the chemical digestion and then the absorption of the end byproducts of the breakdown in the next lecture. So a couple of things just to think about, and while you're waiting, you can actually copy this slide down. So the 6 questions that I indicated from the lecture slides that we've just gone through in the order that I've just gone through, you might want to go back and revisit slide 13, 14, 23, 24. Sounds like we're playing Lotto 649, right? I don't think we're going to win, though. But if you were to go back over the weekend, make sure you're comfortable with those. But again, I want you not to worry so much about your final exam. I think as U of T students, you can't do that, right? It's like an automatic reflex. You kind of have to worry about marks and final exams all the time. But I want you to be thinking about this in a broader context. You know, really, you eat all the time, or you should. I don't know. Maybe if you don't eat all the time, maybe you should think about eating a little more often. But if you're thinking about this, a newborn baby actually is surviving on nothing but breast milk. How does that actually happen? And this breast milk, which we call colostrum, how does it actually survive? Why is the baby's GI tract system, which is actually structurally not any different than ours, how does it actually allow a baby to survive on breast milk? Think a little bit about that, because I'm going to come back to that in the next class. And then I want you to also think a little bit about the different things that I just ended with here. That the nervous system and the different secretions are really not that different and distinct. And which of the different nerves that we've talked about would you stimulate to cause secretion within the gastric system and the duodenal system in some more detail. And again, I'll stick around if you have any questions. Hopefully you don't mind if we leave a little bit early today, and we'll pick things up at the next set of lectures on Tuesday. Sorry, do you guys by any chance have a Facebook study group page or anything? Can one of you let me know how to link this recording to you somehow, and then you can figure out how to disperse it to your classmates?